Leaf ontogeny. Leaf ontogeny explained with the diversity of leaf or development of leaf characters. Diversity of leaf or development of leaf is also called leaf ontogeny. Leaf ontogeny mean diversity or development of leaf. Initially, from the node area of the shoot or branch, from the axillary part, leaf meristem is present. Initially, a leaf meristem. The leaf meristem is also called leaf promeridium. The leaf meristem participated in the periclinal divisions. It developed into three types of initial cells or meristem cells. They are upper initial meristem cells, internal meristem initial cells, lower initial meristem cells. Upper initial meristem cells or upper initial cells participated in the periclinal divisions that develop into upper epidermal initial cells. Upper epidermal initial cells develop into upper epidermis of leaf. Upper epidermis of leaf. And lower initial epidermal cells. Lower initial meristem cells develop into lower epidermal initial cells. The lower epidermal initial cells develop into lower epidermis of leaf, stomata, trichomes, and cuticle. Between these two initial cells, internal meristem is developed. The internal meristem again participated in the periclinal divisions. It can develop into three types of initial meristems. They are sub-upper initial meristem cells, central meristem cells, sub-lower marginal meristem. Sub-upper marginal initial cells or sub-upper initial cells develop into palisade initial cells that develop into palisade tissues. Lower sub-initial or lower submarginal initial meristem cells develop into sponge meristem initial cells that develop into sponge tissue. Between these two marginal initial cells, central meristem is present. The central meristem again participated in the periclinal divisions. It developed into xylem initial cells, phloem initial meristem cells. Xylem initial cells means initial nothing but meristem. Xylem meristem cells, phloem meristem cells. Xylem meristem initial cells develop into xylem. Phloem meristem cells develop into phloem. Finally, from the leaf meristem, complete diversity of leaf characters are developed is called leaf ontogeny. What are the diversical characters here? It is leaf upper epidermis, palisade tissue, xylem, phloem, sponge tissue, lower epidermis, stomata, trichomes, cuticle. These all are developed at the matured leaf. That is the diversity or leaf ontogeny. Matured leaf shows mainly three major parts. Here it is the mature leaf diagram. The mature leaf shows mainly three major parts. They are hypopodium, second one mesopodium, third one epipodium. Hypopodium is also called leaf base. The leaf base is swollen in structure in Fabaceae family, Mimosaceae family. Leaf base small are normal in structure in remaining families. The leaf base or hypopodium mainly helps in attached with the leaf base attached with branch or shoot. Both side of the hypopodium or leaf base, green color small structures are present. They called stipules. If stipules are present, the leaves are called stipulate leaves. If stipules are absent, the leaves are called exstipulate leaves. Above these leaf base, axillary part is present. The axillary part is filled with axillary buds. The axillary bud develop into branch or develop into inflorescence of the plant. Above the hypopodium, long stalk-like structure is present. The long stalk-like structure is called mesopodium or petiole. If the mesopodium or petiole is present, the leaves are called petiolated leaves. If the petiole is absent, the leaves are called sessile leaves. It mainly present between epipodium and hypopodium. Above the mesopodium, expanded leaf blade is present. The expanded leaf blade is called epipodium or leaf lamina or leaf blade. The leaf lamina or epipodium containing base is called lamina base. It present at the lower part of epipodium, and terminal part of epipodium is called leaf apex. Lateral part of epipodium is called margin. Margins are mainly two types, zigzag margins and smooth margins. And on the leaf blade or on the epipodium, veins are present. The veins are look like a reticulate structure. The leaves belongs to dicotyledon leaves. If the leaves are parallel in position, the leaves are in monocot. The veins are contains vascular bundles. Vascular bundle contains xylem and phloem. It is the complete matured typical leaf parts. Now we discuss about the internal structure of dicot leaf. Internal structure of dicot leaf shows mainly three major portions. One is epidermis, second one is mesophyll, third one is vascular bundle. Epidermis is a single layer. It made with rectangular parenchyma cells. It covered with cuticle. The epidermis is again divided into two portions. One is upper epidermis, present at upper side or dorsal side of the leaf. And the second one is lower epidermis. It present at the ventral side of the leaf. Between these two epidermal regions, mesophyll is present. In mesophytes, the lower epidermis shows stoma-like structures. The stoma containing God cells and subsidiary cells. They are mainly helps in exchange of gases like carbon dioxide and oxygen gases during the respiration and photosynthesis. Between the two epidermal regions, mesophyll is present. The mesophyll made with the parenchyma tissue. The mesophyll made with the parenchyma again divided into two types based on the size. One is palisade tissue, second one is sponge tissue. Palisade tissues present beneath of the upper epidermis. They look like a pillar-like structure. They containing large number of chloroplast particles. They are prepared food material by the photosynthesis. And at the lower epidermal region, sponge tissue is present. The tissues are loosely arranged to maintain large number of air spaces. The sponge tissues also contains chloroplast. They are also participated in the photosynthesis. And at the veins of the leaf, vascular bundles are present. The vascular bundles are covered with bundle sheath cell. Below the bundle sheath cells, xylem and phloem tissues are present. Xylem present at the upper epidermal region and phloem present at the lower epidermal region. Xylem mainly helps in the conduction of water and minerals to total part of plant. And phloem helps in translocation of organic substance to total parts of plant. It is the anatomy of leaf of dicotyledons and comes to the monocotyledon leaf internal structure. The monocotyledon leaf internal structure also shows mainly three major parts. One is epidermis, mesophyll, and vascular bundle. Epidermis is a uh, upper and lower epidermal regions. Upper epidermal regions and lower epidermal regions are made with rectangular parenchyma cells. In some upper epidermal regions, some cells are large in size. The cells are called motor cells or bulliform cells. Motor cells or bulliform cells. These cells are having hygroscopic conditions. They are mainly helps in controlling or regulating of water capacity in the leaf. If water scarcity is present in the plant anatomy, the bulliform tissues are loses their water, the complete leaf turn into spring-like structure. It mainly avoiding of transpiration, helps in avoiding of transpiration. The both epidermal tissues are contains or layers are containing stomata-like structures. And between these two epidermal lesions, mesophyll is present. In monocotyledons, mesophyll is only one type of mesophyll, either it is polysac tissue or sponge tissues. They are mainly helps in preparation of food material by the photosynthesis. And veins of the leaf, vascular bundle is present. It's surrounded by the bundle sheath. And below the bundle sheath, xylem and phloem tissues are present. At upper epidermal region, xylem is present, and lower epidermal region, phloem is present. It is the monocotyledon leaf internal structure.